Next, we're going to take a look at neurons, which are kind of the main cell or structural and functional units of the nervous system. They're composed of 3 different regions, and these regions define the functional regions of the neuron as well. Now, the 3 regions are the cell body, also known as the soma, the axon, and dendrites. So those are the ones we're going to look at. Keep in mind, though, that neurons are very long-lived, but mature neurons are amitotic, so they cannot be replaced if destroyed, and they have a high metabolic rate. Neurons are also able to communicate with other cells of the body by transmitting electrical stimuli, sometimes called electrical impulses, which go by the name action potential. So let's look at an actual neuron. So here's a neuron, or nerve cell. You can see we have the big yellow cell body with the nucleus and a nucleolus. We also would have all the general organelles that we expect in cells of the human body. But we also have dendrites, which are a shorter cell projection that is the receptive region of the cell, whereas the cell body, or soma, is the biosynthetic center of the cell. It's receptive in some regards because the signals from the dendrites have to go through the soma to get to the axon. The axon is an impulse generating or conducting region for the neuron. The first part of that axon is called the axon hillock. It's kind of the triangular part that is attached to the cell body. Then we see that axon traveling all the way to the right, and it's going to branch off into axon terminals, which are known as the secretory regions of the neuron, because they often contain either ions that can cause a chemical synapse response or chemicals called neurotransmitters that can initiate a chemical synapse response. So these are things that we're looking at as we talk more about the neuron. So the soma, or neuronal cell body, contains the nucleus. It's the major biosynthetic center of the neuron, and it's a focal point for the outgrowth of the axon and the dendrites. Now, because it's amitotic, it has no centrioles, and it does have some developed Nissl bodies, which is essentially just modified rough endoplasmic reticulum that allows the synthesis of proteins for intracellular use by the neuron. So those processes, the dendrites and the axons, they're found on almost all neurons. Sometimes they're slightly modified in how they look, but they do have different names depending on if we're looking at a neuron in the central nervous system or peripheral nervous system. For example, if you have nerve bundles called tracts, then we're going to be looking at essentially axons that are bundled up together. If you have cell bodies and they're called nuclei, we're going to be looking at cell bodies of neurons, but only in the central nervous system. For the peripheral nervous system, nerve bundles or bundles of axons are called nerves, and cell bodies are called ganglia. So we have a little bit of different terminology there. Now, as we learn about the neurons, we're going to kind of stay chiefly with dendrites and axons initially, dendrites being our receptive region, and axons being our conducting region. So let's look at neuronal dendrites a little more. Now, they're short, tapering, and diffusely branched processes. We know they're the receptive region. They also tend to be the afferent region of the neuron because signals that hit the dendrites are going to travel towards the soma. The soma is almost the kind of integrative system for an individual neuron. So all messages that hit the dendrites are going to be conveyed as messages coming toward the cell body. Oftentimes what the dendrites pick up on are electrical signals that die out over time. They're not action potentials. Instead, the electrical signals transmitted through dendrites are usually conveyed as graded potentials. Again, those are not action potentials. Here in this image, we are looking at a structure for a motor neuron, but mainly the structure we're seeing is the cell body. But many of these processes coming off of it are dendrites, and they have branches that we call dendritic spines. Now, the difference between the dendrites and the axons are not just in function. Dendrites are the receptive region, axons are the conducting region, but axons are also going to be much longer than dendrites. there's also usually only one. They're slender processes, they have a uniform diameter, and they arise from that initial part of the axon called the axon hillock that comes off the cell body. So again, that axon hillock is the initial cone-shaped area of the axon that comes off of the soma or cell body. And so that's going to be the area that initially gets the graded potential that has come from the dendrites through the cell body and finally reached the axon hillock. Only if that graded potential is still strong can it possibly initiate an action potential. Action potentials belong to axons. So there's usually only one unbranched axon. It's the conducting region that leads to the axon terminals. And we've seen this before when you learned about the neuromuscular junction. We were looking at motor neuron innervation by the somatic nervous system of skeletal muscles when we looked at the neuromuscular junction. So here's an image to kind of jog your memory. Here we're looking at the axon of a motor neuron on the right with its branching axon terminals in the middle and its innervation or close association with skeletal muscle fibers, forming the neuromuscular junction. So those axons, those nerve fibers, those conducting regions, with the axon terminals at the end that secrete neurotransmitter, those are going to be really kind of the functional area for communication cell to cell when it comes to a neuron. Because axons, even though they contain all the same organelles found in the cell body, and they are processes or projections like dendrites, they do lack things like endoplasmic reticulum and the Golgi apparatus that are involved in protein synthesis and packaging. So they rely on the rest of the cell to do this for them. So they are instead mainly focused on conducting the action potential from the axon hillock to the axon terminals in order to lead to neurotransmitter release. That neurotransmitter release will either excite or inhibit other neurons or effector cells, like muscles or glands. And most of the signals that we're talking about traveling in axons are anterograde, meaning that the signal is traveling toward the axon terminals. But in some cases, signals can actually travel from the axon terminals back to the cell body. Many of those signals are actually retrograde and are not action potentials. They instead are going to be just stuff like organelles to be degraded, signal molecules, viruses, or even bacterial toxins that are taken in or found in the axon terminals that need to be sent back to the cell body. So we also want to bring back some information we've looked at before, which is myelin sheaths, because myelin sheaths are found on axons, and they increase the speed of nerve impulses. They make action potentials faster. Myelin sheaths are lipoprotein, so they're fatty, they have some protein component, and they coat axons. So they're only associated with axons, and so we would say dendrites are always without myelin or unmyelinated. Keep in mind that we've talked about the glial cells already that produce myelin. The Schwann cells produce myelin for axons in the peripheral nervous system, whereas oligodendrocytes produce myelin for axons in the central nervous system. Regardless of the cell that makes it, myelin sheaths protect the axon, electrically insulate it, and they're going to increase the speed of nerve impulse transmission. Again, that means makes action potentials faster. Here is a Schwann cell that's wrapping around an axon in a jelly roll fashion, and one cell forms one segment of the myelin sheath, and there's multiple segments in peripheral nervous system neurons. That's a little different than what we see for oligodendrocytes, where we can have upwards of 3 to 4 axons have myelin sheath created by one oligodendrocyte. With the Schwann cell, we have these multiple segments of Schwann cells that leave spaces or areas called nodes. They're also called nodes of Ranvier. These are areas where we see a buildup of voltage-gated sodium channels. And this also allows the signal from the axon to not have to go all the way down the axon in what we call continuous conduction. Instead, the gaps in the myelin sheath between adjacent Schwann cells, the internodes, act as gaps for the signal to jump between, from one internode to another. This type of conduction is called saltatory conduction. And saltatory conduction is much faster than continuous conduction. So saltatory conduction is when electrical current passes through a myelinated axon only at the nodes of Ranvier, or internodes. Again, that's where the voltage-gated sodium channels are concentrated, and that allows for the re-triggering of action potentials only at the nodes in this jumping type of conduction. And this happens again only in myelinated axons of the peripheral nervous system, where we have these Schwann cells and the creation of these internodes. And it's much faster than if we had continuous conduction in unmyelinated axons. So let's compare 3 different neurons. For example, we have neuron A that has bare plasma membrane, no sodium voltage-gated channels, and no myelin. So the stimulus is created, but without these sodium voltage-gated channels, cannot be recreated and doesn't fully propagate or travel down the entire length of the axon without dying out. This type of pathway, where we see a signal dying out over time or with a distance, is common in dendrites. And it's going to be more common for graded potentials, not action potentials. If we look at an axon, but one without myelin, but it does have the sodium voltage-gated channels, we'll see slower continuous conduction. In each area where we have more sodium voltage-gated channels, the signal gets to be regenerated, and the action potential gains some strength and can travel further or propagate. And that's enough for the signal to at least make it all the way down the axon. But we have the fastest signaling when we have a myelinated axon with sodium voltage-gated channels at the internodes, or nodes of Ranvier. Here, the myelin keeps the current in the axons going, so the voltage doesn't decay much. There's some insulation here. And action potentials are generated only in the myelin sheath gaps, or the nodes of Ranvier. And so that makes it where it appears that they jump rapidly from gap to gap, but really we just sped up the conduction in these protected areas. So that third example, C at the bottom, with the myelinated axon and the sodium voltage-gated channels in the nodes, that is saltatory conduction and the fastest type of conduction. So keep in mind when we talk about neurons, we can talk about multiple areas and how they functionally work to carry out the processing that a neuron is capable of. And when it comes to axons, we're looking at both myelinated and unmyelinated neurons being able to communicate information cell to cell. Both are present within the central nervous system. But keep in mind that when you have myelin, conduction of action potentials is much faster. And in the central nervous system, myelin sheaths are formed by oligodendrocytes, and in the peripheral nervous system, they're formed by Schwann cells. And we really only see those nodes of Ranvier in kind of that widely spaced manner in the peripheral nervous system. And we see this type of myelination in an organized manner inside of the central nervous system. So much so that we have areas called white matter or gray matter. So regions of the brain and spinal cord where we have a lot of axons covered in myelin usually appear whiter in color because of all of the fat. So those areas are called white matter. Then areas without all of this myelin, mostly where you have cell bodies and dendrites, are called gray matter. In the brain, the gray matter is on the outside and the white matter is on the inside, and vice versa for the spinal cord. So just keep that terminology in mind. You'll learn more about that as you learn more about the nervous system, and later focus on the central nervous system. Next, we're going to be looking at conduction velocity a little bit more and see how we can combine axon diameter and the presence of a myelin sheath and how that affects conduction velocities for the action potential.